All right. So, yep, as Alessio said, I'm Chloe. I currently work at Rothamsted in the UK. For those of you who don't know, though, I'm actually coming to SLU to join you next year from March onwards, working on a fellowship project in collaboration with CIMMIT out in Zimbabwe. So I'm really looking forward to that. But for now, I am talking to you about long-term agricultural experiments. Rather than focusing this talk on sort of one specific piece of research, I thought I'd take a slightly different approach and take you on kind of a tour of past, present, and future research from long-term experiments. I'm going to focus on Rothamsted's long-term experiments because I've been here for 4 years now, so I have quite a good idea of a lot of what's gone on and what's going on with our experiments. And of course, I know at SLU you have lots of long-term experiments as well, but perhaps it's more interesting for you guys to hear about something outside the Institute. So I'm going to run through a whole series of different studies that have touched on interesting elements of long-term experiment research. And the kind of joining thread, I think, between all of these studies is that they underline the unique contribution that long-term experiments can make to agricultural research. So first, they can answer questions that we can't answer using short-term experiments. And then the way we use long-term experiments and research has changed a lot over time. So the questions that a lot of the older ones were set up to address are now not the questions that we answer when we research them these days. So if you kind of continue that thought through to its conclusion in the future, we're probably going to need long-term experiments to answer problems that we haven't discovered yet or questions we haven't thought of yet. So keep those points in mind as we go through, and hopefully by the end of the talk, you'll agree with me if you don't already. So starting in the past, at the beginning of the story, at Rothamsted, we have the Broadbalk winter wheat experiment, which is the world's oldest still running agricultural field experiment. It began in 1843, and it was set up to look at the effect of fertilizers on wheat productivity. So this was around the time that fertilizers were being invented. It is, of course, so old, though, that people didn't really grasp the sort of statistical issues that we now really take for granted. So Broadbalk is neither replicated nor randomized, which makes it sometimes a little tricky to work with now. But at least the fact that it's been going so long gives us some confidence that the differences between plots we see are real differences. So you can see on the right there that Broadbalk's treatments are done in strips, vertically and horizontally. So there's no randomization or anything across that field. But it was actually Broadbalk that helped to contribute to a lot of contribute to the development of statistical ideas around how you tell a real difference from a chance difference. When our statisticians were working on experiments like Broadbalk and saying, well, how do we know if that's a plot effect or a treatment effect? And so a lot of the stats we use today, such as ANOVA, were developed during that period. So perhaps the statistical challenges are a disadvantage of Broadbalk being so old, but an advantage is that it can kind of tell us a story of agricultural development over the last 180 years. So if we look back to when it started in the mid-1800s, and you can see there with those lines, the blue line at the bottom is continuous wheat that's never been fertilized. The pink and the purple line are continuous wheat again that has received farmyard manure or fertilizer. If you then look over to the right of the plot, then the orange and the green lines are where we introduced crop rotations, and that's showing the yield of wheat in those rotations. So I think it's interesting to note that originally when fertilizers were introduced, they actually weren't gaining that much relatively. So they were going from about 1 ton in unfertilized wheat to 2 or 3 tons using either fertilizers or farmyard manure. Very little difference between those two. However, when we get to the sort of 1960s, we see the green revolution happening. We see the introduction of short straw varieties and things like herbicides and fungicides. And we see those yields increase a lot, so that now in the continuous wheat, we're getting around 5 or 6 tons a hectare. And that's obviously because the wheat plants are able to make more use of the nitrogen that's provided to them in that fertilizer, because they're struggling less with weeds and pests and things, and also allocating more resources to grain than to the straw and the old long straw varieties. What's really interesting, though, I think, is that in the rotation, we see those yields double again up to over 10 tons per hectare. And so I think that's really emphasizing the importance of crop rotation and further suppressing weeds, pests, and diseases to allow the crops to really make the most of the conditions that they're in and the nitrogen that they've got. So I guess Broadbalk's comment on sustainable agriculture is that the best solutions are probably not only technological, they're a combination of technology and ecology. So with this, as demonstrated by this history of Broadbalk, we have these really long time spans. So most of the sort of established long-term experiments have several decades of data, and that means that we can study things that take a really long time to stabilize. So a good example of that is soil carbon. Here's a plot from a long-term experiment, a different one, also at Rothamsted, where they started with some areas under grassland and some areas under arable, and then they switched. So they converted some of the grassland to arable and vice versa. And the first thing you can notice on this plot is it takes about 40 years for the soil carbon to reach a new steady state after it's been changed. So if you only did an experiment for 2 or 3 years, you'd have no concept of the overall magnitude of making that change in the long run. Of course, the other thing to notice, though, is where they converted arable to grassland, that's the solid red line, the soil carbon never got as high as it is in the plots that have stayed grassland the whole time. And so that's a suggestion, perhaps, that we can't always put things back the way they were just by changing the conditions. Here's a plot that makes that long time frame even more obvious. So on Broadbalk, in the plots where they've been receiving farmyard manure, that soil organic carbon increase in the soil only starts to flatten off after around 100 years. So keeping these experiments going as long as we can reveal some really useful insights. Climate change is another thing that, although it's happening much faster than we would like it to, you still need quite a lot of years of data to really untangle the underlying effects of a sort of an overall change in climate rather than just interannual weather variation. This paper sort of combines those two ideas, though, and they clustered years by their weather patterns. So they took data on things like, how hot or how cold it was in every month and how much rainfall there was, probably a few other things. And from that, they were able to identify years that had quite similar climate patterns across them. Then when they plotted those against time, they noted that some of those patterns or those weather clusters were much more common at the beginning of the century, whereas others, and these are the ones I've highlighted with the orange circles, these ones became much more common in the last sort of 20 years. And so, I mean, first on its own, that is some evidence that the climate change is driving distinct weather patterns, at least here where we are in the southeast of England. But of course, the next obvious step is to look at the yields in those clusters. So they looked at yields across 3 of our long-term experiments. Those are the different shapes in the plot. And they compared 5 clusters that have been reasonably common since we shifted to short straw varieties in 1968. So that was those 3 clusters that were very common since the 1990s, as well as cluster 2 and cluster 10, which were more common around the middle of the century. And we see, unfortunately, for the future of agriculture, that the yields were generally lower in those clusters that are now more common in the current day. So we're expecting to have a higher frequency of low yielding years for cereals in southeastern England under climate change. So that after we kind of get our heads around the fact that some things take a long time to change, sometimes those changes are small, and so it just takes us a long time to distinguish them from background variation. So often we can use long-term experiments to really help us distinguish, something like the effect of an agricultural practice that might interact with rainfall. So you need to run it for several years to really see what the overall effect of that practice is against the background of weather variability. However, I have a colleague who took that one step even further, and he was interested in not just how the mean changes over time, but also how variance changes over time. So he modeled hay yields on our park grass experiment, so that's a grassland experiment. And he found firstly that the yields decreased in hotter and drier years. But not only that, when you had that yield decrease, it constrained the variance that was possible in the following year. So even if you had a really good year come along on the heels of the bad year, that the hay yield the next year wouldn't increase as much as you would expect for a good year, because it was constrained by the effect of that bad year. So what that unfortunately means is that as we get these hot and dry conditions get more frequent, then even where we still get a cooler, wetter year, the grassland can't recover. So we see greater declines over time. And they've predicted these out into the future, and the declines in yields are not looking good. So we already noticed we had a drought in the UK this year, and farmers were struggling to find enough feed for their animals. And it unfortunately looks like that is going to become a lot more common. But it's not all bad news, fortunately. As a society, we've got some things right. So around the sort of like 1960s to 2000, there was a lot of atmospheric nitrogen pollution. So these are the NOx compounds, and they were typically released from industry, from burning coal, mostly, I think. But we got better at technologies that could take these things out of the emissions from the industry. And so those levels have gone down again. At the time when they were high, ecologists were worried about the result being that nitrogen was being deposited across the UK, and that would select against plant species that are adapted to low nutrient environments. A lot of these plants are quite rare anyway, because they don't survive well in agricultural habitats. So there was a real worry that we might be driving some of these species to extinction. Fortunately, we were able to reduce the nitrogen pollution. And then at Rothamsted, we could again use park grass to see what happened afterwards. Now I have to move my camera thing because it's hiding the plot, right? And so what you can see there with the blue and green lines, which are unfertilized plots, the diversity decreases, in line with that peak of nitrogen deposition. But after the 1990s, when that nitrogen reduces, we actually see that diversity bounce back up again in those blue and green systems. They could also show in this experiment that where they stopped fertilizing some of the fertilized plots, which are shown in the red and the purple lines, then where you see the dashed lines, that's where they stopped fertilizing some of them. And again, they saw that diversity bounce up. So although we have some evidence from park grass that it struggles to recover in the short term from bad weather years, at least we know it can recover from things like nitrogen pollution, which perhaps gives us a bit of hope for its all-round resilience. Ooh, there's a circle. Right, so the last, I guess, point to make about the value of what long-term experiments do that other experiments don't is that when you are applying these treatments for a really long time, then you end up creating quite distinct environments in every plot. And so we can start to look more deeply into those individual plots and try and understand how the mechanisms that perhaps link to things like yield or soil carbon, how they are differing in those different environments that we've created with the different treatments. So a good example is looking at soil and soil microbes. So in this study, colleagues compared treatments in a long-term experiment, which has grassland treatments, arable treatments, and bare fallow areas. So that's where the soil has been kept completely bare for a very long time. And first, they were able to see that this had affected the physical properties of the soil there. So hydraulic conductivity and connected porosity are both things that you want, they're good things. And they were much better in the grassland, a little bit better in the arable, and generally worse in the bare fallow. Much bigger difference between the grassland and the arable and bare fallow though. And this was reflected in the types of microbial communities that were then able to exist in those soil types. So again, we see quite big differences between grassland and the other two, though with some separation between arable and bare fallow as well. And then they found, so this paper was kind of pitched as the idea that soil is it can be seen as an extended phenotype of the microbial community. So because you select for certain microbes through, excuse me, select for certain microbes through certain treatments, you then affect the effects that those microbes have on the soil around them. So they found that by selecting for these different communities, it was affecting the carbon cycling and the nitrogen and other nutrient cycles. And so they found that in the grassland community that had sort of better soil characteristics and those distinct microbial communities, we were seeing much higher rates of carbon being stored in stable forms as microbial biomass or in humus in the soil, and also reduced loss of nutrients, either through leaching or through emissions. But those things were not as good in the arable and the bare soil treatments. Sorry, I'm just going to have a drink. And coughing. Right, so hopefully you've sort of kept up in the last few slides as we've taken a bit of a whirlwind tour from the past and up to the present. And just to really emphasize that we are using these experiments for things that the founders would not necessarily have expected. I mean, whether they even really knew much about the existence of soil microbes back in 1843 is perhaps questionable. But in the next few slides, I want to highlight some studies that are perhaps particularly unexpected. And to do that, I'm going to introduce you to the Rothamsted Sample Archive. And so we have essentially a very large cupboard here where we have stored soil and plant samples from our long-term experiments every year since they were, well, I think every year since they were started. So the oldest ones are from the 1840s at Broadbalk. And a while ago, some researchers were able to look at these samples, plant samples from park grass in this case, and use the isotopes in those plant samples to identify which countries were doing nuclear tests where, in which years, and how far that radiation spread. So you might not expect your average long-term agricultural experiment to contribute to global geopolitics, but they can. Another example that is perhaps not so unexpected to us now, but if you think back to Broadbalk in 1843, DNA wasn't going to be discovered for another 110 years or so after that. And so now that we can go back and use those samples from that time, or maybe not quite that far back, but near that far back, to explore DNA would seem pretty mind-blowing, I think, to the founders. So this is an interesting study where they were curious about how fungicide resistance had spread so fast in the UK after the first fungicides were introduced. And they were able to go back and find out that the resistance genes were always present in the population of this particular pathogen they looked at, just at very low levels, presumably because, pathogens and microbes and so on are always interacting with each other. So it's important for them all to have various genes that help them resist various types of chemistry out there. So we see these genes present in the population. When this particular fungicide was then introduced in the UK, we see this sudden massive increase so that the genes were then present in the majority of those microbes within just a few years. So I think this is a really clear illustration of how selection for resistance works, especially if you rely too much on a single fungicide. I think this was one of the first ones in the UK, so it was probably used, without much variation. Leaving the sample archive now and coming back outside, another sort of unexpected consequence of Broadbalk is that when herbicides were invented, this was quite some time after Broadbalk had started, of course, they made this decision to leave part of Broadbalk herbicide free. So we have a strip of Broadbalk that has never been sprayed with herbicides. And what it's turned into now is essentially a reserve for species that were really common weeds of wheat in the 1950s and are now very, very rare because none of the habitats in the UK are really similar anymore to how people were growing wheat in the 50s. So now we find some of these rather cute, weeds or wildflowers, depending on your perspective, but some of them are barely found outside that strip of Broadbalk. So it plays an important role in weed conservation. Right. So starting to think a little bit about the future. Obviously, I've been talking about how we've been using the same older LTEs to answer new questions. Sometimes now we might start to think about designing new long-term experiments to more effectively address new questions. So for example, there's an image there of Rothamsted's new large-scale rotation experiment, which was started in 2018. So we hope it will soon be a long-term experiment, but at the moment it's just a long-term experiment in training. And that, of course, makes the point that we have to wait a long time to get new answers from new long-term experiments. So in the meantime, we might as well keep going with trying to get answers to new questions out of the old experiments. And so to peek a little, it's not really a little bit into the future now, it's more about the current hot topic. I would say the thing that I'm most excited about are these studies that are bringing together lots of different long-term experiments to answer questions that we can't answer from a single long-term experiment. So you may recognize this figure because it's by several people some of whom I saw in the seminar. And this is a study from SLU looking at using long-term experiments to quantify the benefits of crop diversification to cereal yields, and finding that the benefits were strong and consistent for winter cereals, and initially weak but increasing over time for spring cereals. A similar study looking at 11 long-term experiments across the US found similar results, that when you increase the complexity of a rotation, this is the left-hand plot, then we saw a positive effect on maize yields in most experiments. If they looked only at the most stressful years, so the driest years, then that increase actually got stronger. So the effect of a complex rotation had a more positive effect on wheat yields in dry years. And you can see that if you look at the x-axis of those two plots, the one on the right goes up a lot higher. Here's one from Southern Africa. This is a multi-experiment analysis of 6 long-term experiments looking at conservation agriculture. And here they were able to show that on that plot on the right-hand side, that conservation agriculture practices are more effective under lower rainfall and in sandy soils. And on the left there, they were able to explore how different types of conservation agriculture system performed. And I think notably that light green line is where they have conservation agriculture that includes a crop rotation, whereas the other systems are more just focused on no-till without that much diversity. And then, because I can't possibly go through a whole talk without mentioning my own research once, I have also led one of these multi-LTE studies. We were looking at how ecological intensification interacts with inputs at the field scale. So I've thrown a lot of plots on the slide. Don't look at all of them in detail. Just notice that generally those yield ratios are higher in the light and the green points, and those are sites that had no or little nitrogen fertilizer. So we're seeing that these ecological intensification practices tend to boost crop yields largely, but not entirely, through providing nitrogen to the crops. And therefore we see more of that benefit when we're supplying less of that nitrogen through fertilizers. The one exception is in the top left there, where we found that if you were already providing a lot of nitrogen as fertilizer, then it was actually more beneficial to diversify with non-legume crops, possibly because those crops are able to make better use of that nitrogen, and the legumes don't seem to do very well in those situations. And then furthermore, the yields of the cereals with legumes and high nitrogen were also decreased. So at Rothamsted, we think these multi-LTE studies are really exciting. We think it's amazing to not just be able to say, you know, okay, we're really sure of this result because we can see it across decades, but also to say, okay, we have something generalizable because we're seeing the same kinds of results in long-term experiments, you know, in similar environments across wide geographic ranges, or not. Either way, it's really useful, I think, to help find out how, you know, agriculture on the global scale, what direction it needs to go in order to help address critical problems around food security and climate change. So GLT's been around for a while now and improving slowly. If you haven't looked at it for a little while, I would encourage you to pop back there. There's now, I think, nearly 250 long-term experiments on. We have a mixture of ones where we've had contact with people and they've added their own data. And then we've had a couple of people going through the literature and looking to add other ones, just to make them obvious there, even if we don't have direct contact with the managers at this point. At Rothamsted, we also like to share our long-term experiment data. So we make a lot of it open access through our electronic research archive. So if you have a research question, but you don't have your own long-term experiments and one of ours is suitable, then you know, please do write to us and we'll be happy to share the data. I do believe that Jana in the next talk is going to talk a little bit about some of using some of our data in some of her studies. So that's great to see that. And then I'm just going to end this talk by suggesting that if you're excited about long-term experiments, we're going to have a conference here at Rothamsted next year in June. So please save the date for that. And we're hoping to send out a formal announcement very soon. And with that, I think I'm done. I think that was faster than I thought it would be, which is good.